Hello, you are welcome to Latvian State Forest Research Institute here in Salaspils. Forest area covers more than 54% of territory of Latvia, and forest sector is one of the most important sectors of economy in our country, and our institute makes the theoretical basis for forest sector and forestry. At the same time, we are quite a fast-growing scientific unit with big ambitions to be a stable and serious partner and part of international science communication in forestry. Infrastructure of Silava consists from two parts. One part is forest area, 28,000 hectares of scientific forests located in 5 regions of our country, and as a result of reform of forestry sector in 2014, Forest Research Station is one of our structural units. We manage this forest area together with our partners from Latvian University of Life Sciences and Technologies. More than 1.5 thousand sample plots, and part of scientific infrastructure is located in forests. But we will not go today to forest. We will look the second part, the part of infrastructure which is located here in Salaspils, in our main building, and 3 addresses in Salaspils which are new, renovated houses during last years, and more than 400 new infrastructure units and packages are located in these buildings, and we will show some of them, and my colleagues will tell about the use of this infrastructure in our work. That short introduction from me, and now you are welcome to our institute and continue in laboratories.
The molecular genetics lab has seen a lot of investment in the scientific infrastructure. We see a couple of instruments on the background, and this is a high quality next generation sequencing equipment. We have only 2 instruments of this quality here in Latvia. One is here, and this institute uses this equipment to study forestry and bioeconomy using genetic and genomic methods. We are the best laboratory regarding the analysis of non-model organisms in Latvia, and we have developed strong national and international cooperations, the strongest international cooperations being with scientific institutions from Norway, Sweden and Belarus.
So the Genetic Resource Centre within the Institute is responsible for managing the seed bank where we are at the moment. We have approximately 2,000 crop seed samples which are stored at -20. In addition to managing the seed bank, we are responsible for coordinating all activities in genetic resources in Latvia. So this includes plant species, tree species, as well as animal, fish, genetic resources. The Institute represents Latvia in the European Programme for Plant Genetic Resources, and we also have collaborations both internationally and nationally in terms of using genetic and genomic methods for studying these genetic resources. So the Genetic Resource Centre also incorporates the Molecular Genetics Laboratory. Here we have the DNA extraction room. We also have the main molecular laboratory, and so here we are using these molecular techniques, genetic and genomic techniques, to study all aspects of the work that the Institute's involved with. So this includes both tree breeding, ecology, wildlife management, phytopathology, basically all aspects of the bioeconomy, and also the work that the Institute is doing. And as already mentioned, we have collaborations with other international and national partners, again working with non-model species, working with mainly with wild species, and agricultural species as well as forestry species. Here we have two capillary sequences which are routinely used every day for analyses in our laboratory. Mainly we're using them for microsatellite analysis of tree species, animal species and so on. We also use it for Sanger sequencing.
We are in the forest phytopathology and mycology laboratory. The state-of-the-art laboratory infrastructure provides us an opportunity to integrate into the international scientific research area. Our institution has a long-term collaboration with scientists from all Europe, as well as colleagues from Belarus, Ukraine, New Zealand and Canada. Our team has gained experience about pathogens and pests in abroad studies, international research, as well as coast actions, obtained competencies, knowledges and well-developed infrastructure allows us to include in European research area and fully provide all necessaries for national forestry sector. Besides, our publications and dissertations are prepared in close collaboration with Nordic countries, and also world-leading scientists are supervisors for our dissertations defended here in Latvia and also in Sweden. So these are devices which helps us to make measurements in the forest, and when we have made measurements we can also identify fungi, what kind of fungi cause dieback of the tree or even better helps to the tree. And after that we can make also identification where the microscopes helps us, and when we have isolated pure culture and when we know what kind of fungi it is, we can use this fungi, for example, in biological control agents, but for that we also need to count spores, which we do here under this microscope.
We are here in the laboratory of forest environment, and we are dealing here with multiple issues, starting from impact of forest management on environmental assessment of forest management and finalizing with greenhouse gas emission evaluation of emissions and development of methods. Our priorities and targets are recently quite radically changed, and because we have climate neutrality targets set by, let's say, 1 or 2 years ago, and this is changing quite a bit our research direction from classical to more into carbon turnover and greenhouse gas emissions related research directions. Besides traditional research on increase of carbon accumulation in forests, we are going into more advanced research on soil carbon stocks, carbon flux and greenhouse gas fluxes, particularly from organic soils. And here in this laboratory, a lot of work is done on development of methods to evaluate greenhouse gas emissions from organic soils and carbon turnover in mineral and organic soils. One example why this is particularly important is if we are looking into IPCC guidelines, which are used for accounting of emissions in most of the countries, and for boreal climate regions, the emission factors for forests, organic soils are based on 62 sites, not studies, and for the full temperate region, which is actually all Europe, nearly all Europe, it's only 6 or 8 studies. So it means there's huge shortage of scientific information which is necessary to cover, and that's why it's so important to elaborate country-specific emission factors. And at the same time, our stakeholders are asking us what can be done in our agriculture, forestry to reduce emissions. From that perspective, we are working in 2 directions. One is improvement of reporting, and another is providing guidance for our stakeholders how to manage in future. Another example on emissions, if we are calculating, it's a person in all Baltic states, if we are putting together all the emissions, and organic soils is about 80% of the total emissions, including transport, energy and agriculture and all the rest. So it's very crucial to have these emission factors elaborated in Latvia. There's multiple studies going on. This equipment, this particular is gas chromatograph. We are elaborating a bit on methodologies how to measure greenhouse gas stock contents in small content of in small volumes. You can see these flux. There's 50 milliliters of gas, and it's quite tricky to do measurements in a good, accurate way. We are learning quite a lot from our Estonian colleagues, which are, let's say, developers of these methods in Baltic states. And actually, we are trying in our research to, instead of national research, we are trying to go broader, at least Baltic states level, to have higher value of the research and to have higher, let's say, validation potential for national greenhouse gas inventories when we are applying these methods in practice. So another new thing which we are measuring also by this equipment and by some other, we have GasMet equipment for FTIR equipment for measuring of stem fluxes. Thanks to our Estonian colleagues, we know that there is a significant part of methane emissions are not accounted at the moment because they are coming from tree stems and they are not catched by traditional measurements. So we are going into another direction, which is measurement of greenhouse gas emissions from tree stems, which are very high, especially on forests with high groundwater level and fluctuating groundwater level. So these we are at the moment we are working on an elaboration of the methods for measurements and on development of models which can take in account environmental conditions, 3 dimensions, and fluxes from stems and from soil. We are now in spectrometry laboratory. Here we are determining metal contents and phosphorus content in different extracts. We are using aqua regia extraction using microwaves, mostly aqua regia for metal determination. Then we are determining most of metals using ICP unit from thermo, and when we are determining only potassium and magnesium and calcium, then we are using atom absorption for analysis. Here we are in carbon laboratory. We are determining here organic and non-organic carbon in different samples, starting from water with this unit and finalizing with organic carbon in soil with this elemental analyzer unit. Most of analysis with this unit is done for the international project Torvald, which is aimed to which is Nordic, Baltic, German joint project for elaboration of emission factors and evaluation of climate change mitigation measures for reduction of greenhouse gas emissions from nutrient-rich organic soils. And from interesting results, what we can already say from preliminary studies that we can agree to Finnish colleagues that rewetting in forest lands is not efficient measure. So this is some kind of things which have to be forgotten a bit. And similarly, we can say that from preliminary results that in grasslands also rewetting is not efficient measure. But the project is continuing. We are extending also the project scope by measuring stem fluxes, by determining biomass, by measuring increment of sphagnum, which is very interesting study, not yet done in other Baltic states.
In this laboratory, we are working with remote sensing data. For example, this is our big drone, which is used to get LiDAR data for further terrain processing. And in this corner, you can see the small drone, which can be used for getting those RGB images, which can be used in a orthophoto map and digital surface model estimation. And also we are using this multispectral camera, which can also be used in a different vegetation index modeling. And this is our ground GPS station, which is used for this data combining with the Latvian coordinate system. And our colleague from France, he is doing this data pre-processing in this laboratory. For example, in this computer you can see those RGB image processing, which will be used for estimating the height of the trees. And what you cannot see is multispectral pictures, which, as I told before, will be used in vegetation index calculating, and also with combining with this LiDAR data, it will be used for wet area identification and also for melioration system modeling.
I'm Jordan. I'm a forestry engineer from France. I'm based here in Silava for scientist research, and I'm working with remote sensing and other stuff from mechanical. My field of work is very large.
So here we have a growth chamber, set of growth chambers, altogether 7 of them. We can control a number of conditions in this facility. We can control the temperature with rather high accuracy with the coolers and the central heating system. We can control moisture level, both the air moisture and the soil moisture. And also we can add additional CO2 for complex analysis of tree responses in the climate change conditions. The number of chambers, as you can see here, is important if we wish to analyze tree growth responses to different environments simultaneously, thus obtain the results faster than only with one chamber. Our research here focuses on within tree species response, so genotype differences. We have also hyperspectral cameras taking pictures of those stressed plants, and later on that methodology can be applied or upscaled for the field analysis for larger field test sites. In the growth chambers inside, in addition to other environmental factors, we can control also the light regime. Also, we can assess the tree stress responses in a specific photoperiodic conditions. Also, those chambers are used for development of vegetative propagation technique of the woody plants, and for that also we need a specific controlled set of conditions adjusted for different clones. And when that technique is developed, it can be forwarded for the practical scale applications in the nurseries, private or state sector. Here we have a cross-sectorial research cooperation between the plant physiology and the producers of LED lights, where we assess the impact of different spectrum of different combinations of LED lights on the growth of different clones that are vegetatively propagated. That kind of research issues has been quite well solved in agriculture. However, in the forestry sector with the woody plants, there is a lot of knowledge gaps which we can cover with this type of research. And again, the final idea, besides the fundamental knowledge on the plant physiology, is optimizing the conditions for microclonal propagation and use at the practical settings.
At the moment we are in plant physiology laboratory. We carried out research not only in plant physiology but also in woody plants propagation by microcuttings method. This is biosafety laminar flow cabinets which are used for propagation tree in sterile conditions. Here we multiply hybrid aspen silver birch clonal material and also developing Norway spruce propagation method by somatic embryogenesis. Growing demand of seedling, especially material with highest genetic quality, is basic of our work, and plant producers see possibilities for production of selected high quality silver birch clonal material by microcuttings method, and for Norway spruce clonal material from somatic embryogenesis method. Goal of our work is to identify clonal material with high propagation ability and also developing and finish it rate safe for it, and also propagation conditions. The knowledge will be provided to private sector that carry out industrial scale application for it and plant production. And good example for it is hybrid aspen plant production that has been developed in this laboratory, and after that for plant production in large scale.